So hello everyone, my name is Jennifer, as Leah mentioned, and today I will be presenting some of the slides related to my PhD project. And as you can see from the title, we are studying a technique to treat corneal dystrophies that in most of the cases are age degenerative. And more specifically, the endothelial dystrophies are one of the major causes of blindness. So this means that 40% of the transplantations are due to the corneal endothelial diseases, and this is just in North America. So the corneal transplantation have some side effects and complications, not only in terms of the economical point of view, but also in terms of health, because we can have some cellular degeneration, graft failure, and in the worst of the scenarios for the patient, the rejection of the tissue. For this reason, we are studying in this interdisciplinary project that collects several expertise domains, such as ophthalmology, ultrafast optics, and nanoplasmonics. And this is creating a great opportunity to collaborate with different universities, hospitals, and dependencies, such as the University of Montreal, the University of Alberta, NMIN and NanoCore, to mention some of them. There are other types of treatments for transfection under study, and mostly all of them are related to gene therapy under different approaches, such as the electrical approach, the chemical, physical, virus-based, and optical. I'm not going to go in detail for all of them, but I can show you some of the advantages that are related to it, and is that they are well-known technique. Some of them are under development, and some others are already in the market. But on the other side, we have also disadvantages that are the lack of specificity, the route of administration, toxicity, the persistence of the disease, and also that they are really invasive techniques. So what we are proposing in order to overcome some of the biggest disadvantages of the current techniques is the use of two main tools that are the femtosecond laser therapy and also the plasmonic nanoparticles. So in terms of the femtosecond laser therapy, we have some advantages, such as the technique is well known, it's already in use. We can do optoporation and transfection. We are at the therapeutic window since we are irradiating at 800 nanometers. We have a high precision laser beam since we can focalize the laser, and that becomes in a non-invasive technique. On the other side, talking about the plasmonic nanoparticles, we are using specifically gold, because this has been proved to be biocompatible. We have also photostability, and we can use less energy in terms of the laser because we want to reduce the side effects. Also, we can use them as a surface markers. So if we move to the principle, we have to picture, let's imagine first of all that this is a cell that has been cut. So this is a transversal cut of a cell. This is the cell membrane, the cytoplasm, and the nuclei is somewhere here. So we first attach the functionalized nanoparticles, gold nanoparticles. The second step is to irradiate with the defined wavelength, and the laser is going to be absorbed. The energy is going to be absorbed by the nanoparticle. This is going to create a near field enhancement that will lead into a plasma generation. And this plasma generation subsequently will be a plasma-mediated nanocavitation. That means that we are going to have a bubble, produce a nano bubble that will explode at a certain point and produce a shock wave. So this shock wave will allow us to have a hole in our membrane. And once this hole is produced, we can introduce exogenous molecules such as DNA, mRNA, or any other molecule. And usually what we do is to attach fluorophores also, so that we can realize, we can see that the fluorophores are getting inside of the nuclei of the cell, because we are going to receive some fluorescent signal from the cells. In terms of the novelty, we are offering a combined effect of the laser and the functionalized nanoparticles. And this is because we want to offer a high efficiency treatment, a non-viral gene transfer technique, and a double specificity, meaning that we are using the laser and also the nanoparticles. At this point, I would like to also highlight that our group has a patent related to the delivery of exogenous molecules in the eye. So this brings to have tests and controls that we have to do. in terms of optoporation and cell viability. That means that we are opening some pores on the membrane, but the membrane of the cell have to be resealed. And this is related to the viability of the cells. That means that we have to make a trade-off between the transfection rate and the cells that are still alive. On the other side, we are also doing some sort of toxicity tests that are related with the laser and also with the nanoparticles. The main idea is that we are testing the two tools separated so that we prove that none of them are causing any effect alone. But when we combine them, we are achieving this effect that is the optoporation and subsequently the transfection. So in terms of the radiation, the first step is to incubate the nanoparticles on the cells, and then we add the molecule that we want to transfect or internalize. After that, we are ready to use the laser, so that means irradiate at 800 nanometers, and the nanoparticles are going to be absorbing the plasma-mediated the energy, so we will have an effect of plasma-mediated optoporation. This means that we are going to have localized pores on the membrane, and after that, we can see that by diffusion, the molecules are going to be internalized and then bound with the DNA of the nuclei, so we can observe the fluorescence. So more in detail, I'm going to show you some preliminary results and explain you how we do this. So the first step is to stain all the nuclei of the cells. And now let's imagine that this line that we have here, this rectangle, it's our laser. So our laser is going to irradiate in a certain pattern that, for instance, in this case is a line. And by adding another type of dye that in this case is red, we are going to see the contribution of the cells that have been irradiated just in red. After that, we need to prove that our cells are still alive. So we are irradiating, we are adding, another dye that is green in this case. When we do the combination of the three channels, we can have a general overview of the plane of the cells that are irradiated and optoporated. With this technique, we have achieved the 70% of internalization efficiency, and we are working to increase this efficiency. And also we are working in terms of the cell viability to keep most of the cells alive. So this research is also part of an NMIN funded project entitled Site-Specific Laser-Mediated Gene Therapy for Corneal Endothelial Diseases. That means that we have some future work to do. And this is related to ex vivo work. So that means that we are going to use mice models. We will internalize mRNA molecules in cells and tissue. And finally, our goal is to do in vivo therapy with the same mice models used to study the corneal diseases. This proof of principle research highlights that the treatment offers negligible toxicity. That means that our laser is safe for the surrounding tissue, and also that our nanoparticles are a safe tool that we can use in our next stage that will be in vivo. The procedure is less invasive than the current available techniques, since there are no tissue transplantation. And also we are using safe and approved techniques that are now combined to enhance the effect of the laser therapy. So finally, we are aiming to provide a safe and novel alternative for the treatment of corneal dystrophies. And with this, I will conclude my presentation, and I will be happy to take any of your questions. Thank you.
Thank you very much, Jennyfer. So from my understanding, the femtosecond lasers are used for performing eye surgery. How safe is it for that range of power when used for this technique?
Okay, thanks for your question, Leah. It's a good question. The idea of combining this femtosecond laser with the plasmonic material is exactly that we can use now less energy, because the effect of the laser, it's going to be enhanced by the nanoparticles. So like this, we can ensure that our tissue is going to be totally safe when we are using the plasmonic material.
Great, thank you. And then could you clarify what you mean by photostability when you're talking about the plasmonic nanoparticles?
Yeah, totally. So photostability, we are talking about this because we are using plasmonic material. Sometimes when some people is using biomarkers, they are using fluorophores. So this leads into sort of photo bleaching. In our case, we are not depending on any source of light. We are just shining the light to visualize the scatter of our nanoparticles, and that won't be dimmer by the time.
Great, thank you.